Okay, so I'd like to welcome everyone to Youth Neuro Australia's Brain Awareness Week webinar, Demystifying Brain Research. My name is really excited to have Dr. Suraj Samtani, a Postdoctoral Fellow at the Centre of Healthy Brain Aging, or CHeBA, join us for a webinar to demystify brain research. Just please note that this webinar is being recorded, so participants shouldn't be able to unmute themselves at the moment, but if you are unmuted at the moment, please mute yourself. First, we would like to acknowledge the traditional owners of the land where this event is being hosted from today, the Walla medical people of the Eora Nation. We pay our respects to elders past, present, and emerging, and welcome all First Nations people joining us today.
So CHeBA, the Centre for Health Brain Aging, is a premier research institution at UNSW Sydney, striving for advances in brain aging and age-related brain diseases. So Dr. Samtani is a study coordinator for the social health and research in the dementia patient journey, so which is also otherwise known as SHARED project, which he will be discussing during his presentation today.
So Dr. Samtani will be talking about what brain research is and why it is important to all of us, not just those interested in science. And he will also discuss the research that CHeBA undertakes and the methods, findings, and future directions of the SHARED project.
So at the end of the presentation, there will be time for a Q&A section where you can ask anything related to research, brain research specifically, dementia, and also more. So if you have any questions throughout the talk, please type them in the chat, into the Zoom chat, or write them down and save them for the end. So without any further words, please take it away, Dr. Samtani.
Thank you for that welcome. As I mentioned, my name is Suraj Samtani. I'm a postdoctoral fellow at the Center for Healthy Brain Aging. I'm really happy to speak to you today about what brain research is, how we use neuroimaging, and how we use this data when we look at dementia and predicting dementia in the population. So as we get started, I'd like to begin by acknowledging the traditional custodians of the land, the Darug people of the Eora Nation, and acknowledge elders past, present, and emerging. So a little bit about me to get started. My personal pronoun is he or his. Home to me is a few different places. It's Mumbai, where I was born, Auckland, where I did high school, and Sydney, where I did my undergraduate degree, postgraduate degree work, and research. So I did a PhD in clinical psychology from UNSW Sydney. I specialized in repetitive thinking processes and how they maintain depression and anxiety. And I worked for several years in clinical psychology before joining the Center for Healthy Brain Aging in late 2019 as a postdoctoral research fellow. So today I'm going to talk to you about a range of topics. Firstly, why should we care about brain research? Why is this important for us to look at? Secondly, what are the different types of neuroimaging and how they actually work under the hood? And then I'm going to talk about dementia research and the role of neuroimaging in not only diagnosis, but also in research. To demonstrate how we use brain data, I'm going to talk a little bit about the SHARED project that I coordinate at CHeBA, where we look at social connections and why they're important for maintaining a healthy brain. So what's the connection between social interactions and brain health is our key question in this project. And how we use brain data to answer this question is what I'm going to be talking to you about today. And finally, I'll end with why you should go into brain research or why you should care about brain research, whatever field you're from. So let's get started. So why should we care about brain research or brain imaging? Well, we know that neuroimaging now is where biology or chemistry was in the 1800s. It's the final frontier of science in that there are many new technologies, many new discoveries just waiting to be made. And we get to interact with experts from all over the world, people who are experts in different fields in making the images better, more accurate, and helping us to help other people. So it's very exciting, and we have a lot of discoveries in front of us. That's what made me go into this field, because I felt like there was a lot left to discover. So let's talk a little bit about what imaging is. Now, you may or may not have a background in neuroimaging, so I'm going to take you from the ground up here and explain what the different types are. So this picture here is of an MRI machine. Now, it's a giant magnet. Now, maybe you've seen shows like House or other shows where people walking wearing, glasses or belts or watches. Now, that would not happen in real life because it's a giant magnet. You would literally fly across the room and get stuck to it. So the next time you're watching these shows, just keep an eye out for what they show you on screen. When we think about imaging, we've got a bunch of different types of techniques that we can use. You may have heard of some of these, like EEG or PET scans or MRI. Now, these different types of scans use different techniques and give you either information about brain structure or brain function and how activity moves across the brain over time. Now, I'm going to tell you a little bit about each of these techniques so that you understand them a little bit better. So here's EEG. This is where they put a cap on your head and there's different nodes. So there will be nodes on your frontal lobe, which processes reasoning, on your occipital lobe, which processes vision, on your temporal lobes, which processes memory, and your parietal lobe, which processes spatial awareness. So looking at the data from these nodes, each node represents a wave here. So looking at the waves, I can tell whether you show more activity in your frontal lobe or your temporal lobes or your occipital lobe. The great thing about EEG is really fast to respond to whatever stimuli we show someone. So it's in fact the fastest technique. What it's not very good at doing is showing you what the brain is doing under the scalp. It just tells you the total sum of electrical energy that reaches the scalp, which is picked up at the node. So it gives you temporal information, that's information over time, and structural information about where the activity is. And it's a rather crude measure of activity, but you can use it in very interesting ways. So for example, here's brain scan of someone who has herpes and is experiencing an epileptic seizure. So you can see the nodes here are placed on the right hemisphere, and you can see an electrical storm happening, and that's what a seizure is. So you can see things like this using EEG. Next, let's talk about CAT scans or CT scans. So these scans are fairly widely used in hospitals and research clinics. So basically they're just x-rays of your brain, in case you were wondering what they are. And the computer basically enhances these images, and you take an x-ray at every level, and as you slice down the brain, you get a collection of images like this, which show you what the brain looks like at different levels. And this tells you the structure of the brain and what it looks like. It's the least expensive method. So if someone you know has ever been in a car crash and been rushed to a hospital to check what their brain looks like, they've probably had CT scan done. So you might notice these gaps here in the brain. So this is called the ventricles of the brain. Now they are the plumbing system of your brain. They contain a fluid called cerebrospinal fluid, which brings in nutrients and takes out waste from your brain. So you can see here in schizophrenia, we found large ventricles that keep expanding. So they're losing a lot of brain matter. And we see things like this in Alzheimer's and dementia too, which I'll come to later. Now, for those of you who like pets, there are also PET scans. Sorry for my lame jokes. So PET stands for Positron Emission Tomography. And basically this gives you a picture of what the brain looks like across time as someone is looking at something or thinking about something or doing something. So you give them radioactive chemicals, which then go into the brain, and you can see them glow using the scan. So they're basically X-rays as well, but glow in the dark X-rays. So here you get red and orange showing you more blood flow. So basically we're assuming that wherever you have more blood flow in the brain, you're using that part of the brain the most. Now, there's a common myth that people only use 10% of their brain. As you can see here, that's not really true. We're using a lot of our brain all the time.
Because of COVID.
So how can we use this? So we can use this to study different neurotransmitters like dopamine and how it moves across the brain over time. Now, one of my colleagues in the psychology department at UNSW did her PhD looking at cognitive evolutionary psychology. So this is how we develop skills that our ancestors used. So things like flint knapping of stone tools. And she had expert flint knappers and novice flint knappers. And what she found was when you're a novice flint napper, you're trying to learn this new skill, you have to use a lot of your frontal lobe, which does reasoning and planning to learn this new skill. But in expert flint nappers, that activity had moved to the cerebellum, which is at the base of the brain and deals with automatic functions that you can do, fairly easily, like driving a car, riding a bike. And that's what expert flint nappers showed, that they could do that on autopilot and the cerebellum lit up in the brain scans. The next technique, something you probably heard a lot about, and this is MRI, which stands for magnetic resonance imaging. So this uses a range of techniques like magnetic fields, radio waves, and enhancement of these waves using the computer. And MRIs give us much better detail and structure, much clearer pictures of the brain compared to CT scans. And they're getting better and better. The images that we can look at nowadays are much sharper than what we had access to 10 or even 5 years ago. But the problem with MRI, it's just a Kodak moment, it's just a click, one moment in time. It doesn't tell you how activity in the brain is moving over time. And that's where the next technique comes in. That's functional MRI. So fMRI uses several variations on MRI technology, but basically what it's looking at is blood oxygen levels. So assuming that wherever you see high levels of oxygen in the blood, in the brain, that's the part of the brain being used the most. So you can look at several things here. You can look at function and structure over time, how things are changing and moving. And one way this has been used is to look at cocaine craving and the type of activity you see in the brain and how warped it is when someone is experiencing that craving. It's quite interesting. Now we come to my favorite method of brain imaging, which is diffusion tensor imaging. Now this is the only technique that lets us see the white matter of the brain. For those of you who haven't done neuroanatomy, think of the cell bodies and the wires connecting them. The wires connecting the different cells in your brain, the pathways, this is the white matter in the brain. And this is the only technology that lets you see that. So if you close your eyes and you imagine yourself flying over a city in a helicopter, looking down at the city, all the other imaging techniques would only let you see the buildings, but none of the roads. This is the only technology that lets you see the roads connecting the buildings. Why this is interesting is because let's say if someone's been in a car crash, you rush them to hospital, give them a quick and cheap CT scan that's easy to do. All the structures look fine. You say, all right, you look okay, why don't we send you home? But what you won't see in there is that sometimes a car crash or an impact or trauma to the head might cause shearing, where the wires connecting the different parts of the brain have been cut because of the impact, and that's going to cause problems later down the track. So this is the only technology that lets us see the axons or the white matter in the brain. Now, it's also the most colorful psychedelic one. If you're wondering what the colors mean, they actually image the movement of water inside the brain. So you can see here the corpus callosum, which is the bridge connecting the two hemispheres. That's the pink color represents water moving from side to side, and green represents water moving front to back, and blue is top to bottom. This is being used a lot now in dementia since it's become available recently to look at what's going wrong with the connections in the brain in dementia. So when we talk about dementia, it's important to know what we're talking about here. It's actually an umbrella term that corresponds to a range of clinical syndromes, all of which have something in common. And Alzheimer's is the most common type of dementia. So the symptoms or signs that you might see in dementia include memory loss, mood changes, changes in behavior, thinking, reasoning. And a lot of people think that dementia is just a normal part of growing old, but actually most people grow old without ever getting dementia. It's only a small proportion of people which will get this disease. So the main concepts I need you to know about dementia is that it's an umbrella term used to describe a range of clinical syndromes, and Alzheimer's is the most common cause. Some of the causes are reversible, but these are quite rare. And the appropriate language to use for people experiencing this condition is people living with dementia. So we don't use the word demented because that's not respectful, and I want you to remember that as well. So in talking about the research that we're doing at the Center for Healthy Brain Aging, I'll talk about three distinct groups. So first group is cognitively normal. There's nothing wrong with their brain or their function. Everything is fine. The second group is starting to experience some problems with their memory or other cognitive function. It's like their behavior is starting to change. So they don't yet have dementia, but they're starting to notice some changes, and we call this group mild cognitive impairment. And obviously the group that has dementia is experiencing significant changes in their memory or their thinking. If you want to be a nerd about this and look up the DSM, or the Diagnostic and Statistical Manual, these are the terms that you will see in the DSM, which I'm giving you in brackets here. So why should we care about dementia? Well, 500,000 people in Australia living with dementia at the moment. And by 2050, it's going to be about 1 million people, and it's going to overtake depression, heart disease, and cancer as the biggest cause of disability. That's because of our aging population. So the estimated cost in Australia is about $6.6 billion a year. And so that comes down to about $18,000 or $18 million a day, and $12,500 a minute. So if you want to try this activity, I'm going to give you a few seconds. I want you to get your calculators out. We've been talking for about 22 minutes. I want you to work out how much dementia has cost us in the last 22 minutes. Ready, set, go. I'm going to work it out as well to see.
Dr. Suraj, I think there's an answer. So it is like 251,000 and like 140.
That's pretty close to what I got, so well done. That's a lot of money, isn't it? You could buy a house like somewhere in I don't know, regional New South Wales for that, or in a land far, far away. So how can we use imaging to diagnose dementia? I have a video here which I want to share with you. It's just a brief video to explain what we do with imaging. So let's have a look.
By your late 70s, most people start to have mild memory loss.
Just a quick sound check. Is that all right?
Yep,
Great.
thank you.
Craniocervical junction.
It's a normal part of aging, says CDI neuroradiologist Ricardo Sanchez.
Well, a subtle sign would be an inability to recognize a person that they should recognize, or it may take them a little bit longer to identify that person.
According to the Alzheimer's Association, 5.7 million Americans are living with Alzheimer's. By 2050, this number is projected to rise to nearly 14 million as the number of older Americans rapidly grows.
Is that okay?
Leading more and more people to their doctor looking for a diagnosis.
I'm going to start the first scan.
An MRI of the brain can help with that diagnosis. The images help your radiologist immediately rule out certain diseases.
I want to make sure that I'm not dealing with a cerebral infarction, a stroke, old trauma, an unexpected brain tumor.
Then advanced software compares your brain volumes to a large database of gender and age-matched patients.
Central brain atrophy.
The comparison can help you know whether memory loss is related to Alzheimer's.
This is important because, as we all know, there is a tremendous amount of research, massive efforts in the United States and other countries to try to understand and to treat Alzheimer's.
New medications hold the promise of slowing the disease, and an accurate diagnosis can help families make important decisions about care and quality of life.
I'm glad that we are able to make a contribution to a patient's diagnosis, to be able to reassure the clinician that this is the disease process that we think we're dealing with.
All right. So I hope you found that interesting to see how we would apply neuroimaging. In fact, in the diagnostic manual, they've made neuroimaging a compulsory part of diagnosing a range of dementias now. So that you're not just doing a memory test for this person, but you're also confirming brain pathology using these scans. So I want to follow this up with a video that shows you what dementia actually does to the brain. So I'm just going to show you the first minute and a half. It's quite brief, but I hope you find this interesting.
Over a century ago, a German doctor called Alois Alzheimer spotted anomalies in brain sections from a patient with dementia. Ever since, people have been studying the strange plaques and tangles that he saw, in the hope that we could one day understand and cure what is now known as Alzheimer's disease. Plaques are insoluble deposits of a peptide called amyloid beta, or A-beta. They're formed when a protein called amyloid precursor protein is sequentially cleaved by two enzymes, beta and gamma secretase. Other molecules are generated by this cleavage and may play a role in the disease, but A-beta is the main culprit. A-beta tends to misfold and become sticky, eventually clumping together to form soluble oligomers. Some of these aggregate into large insoluble fibrils that deposit in the brain as plaques. The oligomers come in several forms or species. We don't know exactly which species are toxic, but research shows that they weaken communication and plasticity at synapses. This could be what stops the brain from forming or retrieving memories.
So that's all I want to show you for now, that these proteins get severed and they misfold and they clump together and block the signals from one neuron to the next. And we know the brain has a use it or lose it rule. So when you can't talk to the neuron next to you, that neuron gets left out because it hasn't fired, your brain prunes it away and kills that cell. So this is how, with the buildup of these plaques and tangles across the brain, you start to lose tissue in the brain. So what do we do at the Center for Healthy Brain Aging to figure out what's happening with dementia, trying to prevent it, trying to cure it? We have a range of research groups at CHeBA. So we have epidemiology, which I'm part of, looking at how we can prevent dementia. We have the neuroimaging arm, we have the neuropsychology arm, we have genetics and epigenomics, neuropsychiatry, and omics like metabolomics, and nanoparticles. So it's really interesting to see what we're going to discover in the next few years, bringing together experts in these different areas. So I'm going to talk a little bit now about the research that I've been doing with my team. So I'm working with Ashley Stevens, who's a research assistant, and the two co-directors of the Center for Healthy Brain Aging, Henry Burdatti and Parminder Sachdeva. And together we're looking at the role of social interactions in maintaining healthy brain function as we get older. So this grant was awarded by the European Union JPND, which is the Joint Program for Neurodegenerative Diseases, and we're being funded locally as part of an agreement through the NHMRC. So this grant is for 2.5 million euros over 3 years, and we have 7 partner universities that you can see here on the right, including UNSW. So what we're looking at is we're taking data sets that follow people longitudinally over several years and looking at the data from multiple countries around the world and combining them to answer important questions about dementia. So why are we looking at social factors or social interactions? Well, we know that it's one of the 12 modifiable risk factors for dementia and accounts for about 4% of preventable cases. So that would mean if we prevented people from experiencing social isolation, we would prevent about 5 million cases by 2050. That's sort of equivalent to the population of New Zealand currently. So that's a lot of people. So when we talk about social factors, what are the factors that we measure? They're called social health, and there's a range of measures that we can use. So we can use loneliness or isolation. We can look at the quality of their relationships with family and friends, with network size. That's how many people they know across their family and friends, whether they have social support, whether they participate in activities, and whether they have autonomy. Can they go to events on their own? Can they continue to participate in the workforce? So why is this important for dementia specifically? We know from a lot of research that being married, having many social contacts, having a high level of social activity is protective against dementia. We also know that people who have more social support tend to maintain their thinking and their memory down the line. And that this might be stronger for women compared to men, this effect of social support. And we know that people who feel well understood by others around them also show less decline in their memory 20 years later. So we know that social relationship quality and quantity are both really important, and that's what we want to look at. But what we don't know is what are the pathways between having lots of social interactions and good social interactions and maintaining good cognitive health. reasoning, memory, decision making, all of that. Well, we're really interested in the shared project in this aspect, which is brain reserve. So brain reserve is when someone has lots of connections in their brain. Maybe they've had many years of education, or they've been doing lots of interesting activities throughout their life, so their brain is very connected. And what you see is when they start to lose some routes in the brain, they can still use alternative routes to think about something in a different way. So our question is, does having lots of social interactions help you maintain that brain reserve and help you keep those connections? So our questions are, what are the pathways that link social interactions with cognitive health? And what's the relationship of social interactions with brain reserve and ultimately cognition? And what happens to people's brains and to their social interactions as they develop from mild cognitive impairments to getting full-blown dementia later down the track? So our grant and our teams are structured in that we have different countries looking at different things. We have a country that's building theoretical models, another that's mapping trajectories over time, another that's looking at people going from healthy to having some memory complaints. And our team at UNSW, we're looking at people who have some memory complaints and following them up to the stage of dementia. So our team aims to compare people who have mild complaints with those who have dementia, look at the bidirectional relationship between social interactions and cognitive health, and look at how variables mediate this relationship, especially brain reserve. Does it come somewhere in the middle? More social interactions equals more brain reserve equals more cognitive health. That's what we're trying to answer here. And we're trying to discover novel biomarkers, so maybe social factors combine together to influence your immune response, and then that leads to changes in the brain. So what we're hoping to do is identify what are the key social factors and how this relates to changes in the brain over time, and what are the mechanistic pathways that occur in the brain that result in changes in memory as a result of social isolation, so that we can make solid, concrete recommendations for interventions. So in our first study that we just completed, as part of this project, we created three groups. So we had two groups who had mild cognitive impairment at baseline. One of them remained fairly stable and they were healthy or had mild complaints at the final wave. The second group started off with mild complaints and ended up getting dementia, and the final group already had dementia at baseline. So we looked at studies of aging and longitudinal studies, and we found 12 of them that met our criteria. I won't go into this in too much detail. I'm just going to talk about what we found and what data we had. So we had data from Australia, Singapore, South Korea, China, the Central African Republic, the Republic of Congo, Greece, Germany, Sweden, UK, Brazil, Puerto Rico, and the US. So quite a global sample. So what we found is that at baseline, people who had symptoms of dementia tended to interact much less frequently compared to those who had mild cognitive complaints or mild impairments. So this was not surprising, because you'd expect people with dementia to be interacting less often because of the difficulties that they're experiencing. And then when we compared the two groups with mild cognitive impairment, we found that the group that declined over time were less likely to have someone to confide in, someone they could talk to about their difficulties over time. So maybe it's something about having someone to confide in and having these frequent social interactions that helps keep our brain healthy. So Now we're going to do the next step of looking at brain data and how social interactions impact the brain. What do we know from the studies that have been done on this topic so far? Well, we know, one, that people who got tangible social support, that's support with things like showering, eating, shopping, going to family events, those people with a lot of support show greater brain volumes in the temporal lobes and the frontal lobe. So these are parts of the brain involved in memory and executive function, which is planning, reasoning, decision making. However, the studies that have been done so far only used healthy people and were cross-sectional. So what we really need to look at is what happens in the brains of people who have these mild cognitive complaints or dementia, and also track the changes over time. The other interesting study that's been done already on this topic found that people who have high contact social roles. So for example, they don't just like meet lots of people, they meet some people lots of times, quite frequently. And these people showed that they had higher brain volumes in parts of the brain that are already linked with better memory. Again, the same problem in these studies. Healthy people, cross-sectional design. So our questions that we want to answer are, in people with cognitive impairments, can we see differences in the brains of people who stay socially active versus those who are isolated? And how does their brain then change over time in response to how much they have been interacting? And to answer that question, we're going to use a different data set, which is quite rich, which is called the UK Biobank. It's a large biomedical database, which has data from 500,000 people. And about 50,000 people out of that have brain scans available now. It took me a whole day just to download their data and a whole day just to unpack it. And if you want to download the brain scans, it takes about 3 months to sit there and download their brain scans. And that's just a snapshot of their brain at one moment in time. If you want data over time as they do a task, you'll be there downloading for a few years at least, I imagine. So what data do they have? Firstly, they have structural MRI, so that's volumes of different regions of the brain. And they also have data from diffusion tensor imaging. Remember the psychedelic colorful imaging that we spoke about? That shows you the wires connecting different parts of the brain. Now what happens when those wires get damaged, that damaged spot shows up as bright white spot on these scans, and that's what we call white matter hyperintensities. So that's what we're going to be looking at. So our next steps are going to be about looking at brain volumes and these white matter hyperintensities in people who show different cognitive trajectories over time, people who stay healthy or people who decline into dementia. Another thing that we want to do is look at specific brain regions linked to social interactions. For example, we expect that people who stay social will show greater brain volumes in the orbital frontal cortex, that's over here, which is linked not only to staying socially active, but also to reasoning and personality. Also, we expect that they'll show higher brain volumes in the hippocampus, which is a part of the brain that stores memory, because we know that staying socially active means you're talking, you're using language, you're using your memory. That keeps your hippocampus going. The other part of the brain that we're really interested in is the amygdala, which stands for almond. It's next to your hippocampus, and it is the fear center of your brain. So this part of the brain controls your fight or flight response or your anxiety response. So we know that people who've been isolated, they go into a defensive state where they experience chronic stress because of their isolation. And what we know chronic stress does to the brain is it causes shrinkage in the hippocampus and too much activity in the amygdala. And what we want to do after that is look at longitudinal effects, is looking at how these social interactions at baseline show up as changes in the brain 1 or 2 years later down the track, and how that then impacts cognitive function later on. So that's what we hope to answer in the coming year with this project. So to wrap up, I just want to talk to you about why you should go into brain research or care about brain research. Firstly, there's many discoveries to be made. Secondly, there are intersections with lots of different fields. So maybe you go into this as someone who works with brain imaging directly, or maybe you're just broadly involved in health and get access to brain data, or maybe you work as a statistician or look at population health or computer science or engineering. You can come at this from many different angles, and I hope you've taken away from this talk why neuroimaging is so exciting and important, because it's also quite rewarding, and it also helps us to help other people by making these new discoveries. So that is it from me. So you can contact us at CHeBA. Here are our details. I'm just going to wrap up and stay online for some Q&A.
Yeah, thank you so much for that incredibly insightful presentation, Dr. Samtani. So as Dr. Samtani said, we're now accepting questions in the chat for the Q&A, and you can also request to be unmuted if you would like to ask a question over voice chat. Okay, so Leila's first asking, are dementia patients who lack social interaction are more likely to have faster disease progression?
That's what we've seen in some of the research that's been done so far, is that people who have very few social interactions tend to decline faster. And we hope to confirm that, replicate it, and see if that gets replicated in our global sample from 12 different countries.
Okay, so thank you for that. So there's another question from Rohit. So he asks, like, can we quantitatively assess quality of social contacts rather than just number of contacts?
Yes, that's what we hope to do as well. So a range of studies that supply data to us have data on relationship quality. So how positive or negative their interactions are with those around them, whether they feel belittled by the person looking after them, whether they feel pressurized or controlled by the person who looks after them, or whether they have a very happy, positive relationship and the person who looks after them, their care partner, encourages them to do activities.
Zara asks, how have changes in technology since you were a student enable your current research?
So when I was a student, we didn't have access to DTI. Only 4 labs at the time had access to it. So it means that we can now look at the connections between different brain regions using that technology, which is really exciting because I suspect that the connections break down before the cells die themselves. So we're likely to spot dementia earlier than anyone's been able to spot it before.
Okay, thank you. So there's another question from Mary. So like, she's asking, what is one of the main challenges that you have found in your work?
One of the main challenges that I found is just the size of the data that's available. So when looking at the brain data, we've got access to 18,000 variables. So it takes a long time to run descriptive statistics and data cleaning and data checking before you even get to analyzing that data, because the data sets are humongous that we can only open about 100,000 people's data at a time, otherwise the software crashes.
Nick would like to know, what are the future plans for your findings and what are the clinical applications/implications?
So what we hope is in our next studies to look at the changes in the brain that result from having high frequency or high quality social interactions, and exactly what type or how much, And what we hope to do is inform healthcare and policy to recommend, if you have someone in front of you at the doctor's office who presents with some memory complaints or other cognitive complaints, well, it's good to just recommend social activity, but what type and what variety and how much do they need? That's really the key question that we want to ask.
So Hui has another question. So it's about like, will growing in unfavorable environment, for example, like noisy and full of screaming noise, like cause the failure in brain development and leads to dementia?
So growing up in a noisy environment, did I get that right?
Yeah, like in a noisy and full of screaming noise environment, so will it cause the failure of brain development and also leads to dementia?
Well, we don't know if it will link to dementia directly because the studies that we look at looks at data from middle age to old age. So often we don't get data because there are no 70 or 90-year-old studies that are out there. There's maybe 30-year-old studies that are out there. So what we know from studies of childhood is that people who grow up in an environment where there is trauma or domestic violence or mental health issues that impact the child, in that the child's being neglected or mistreated, that child's brain is not going to develop as well as other children's.
Rohit has another question. How do you adjust for better cognitive function, allowing for social interaction, which could confound results?
Sorry, I didn't get that question. Voice broke up there.
Yeah. How do you adjust for better cognitive function, allowing for social interaction, which could confound results?
So if I understand your question, you're asking about how we can control for important variables like cognitive function. So what we do is we take someone's age, sex, and education into account when we look at models. And we also control for their baseline level of cognition when predicting or looking at their final cognitive levels.
Okay, so Lilo also has another question. So it is like, what will be or has been the hardest step in the shared project timeline?
The hardest step, I think, has actually been trying to work out what every team is going to do, because you have such a big idea and you've got to think about, well, what's our team going to do that we're experts in, and what's your team going to do that you're experts in, and what databases do each of us have access to that we can then analyze, and then figuring out, are these databases actually good quality? Do they have enough social variables in there for us to look at? So that took us a solid 6 months to look through every question in every study to see which ones will be useful to us.
Jack asks, would you say that social isolation is a lasting influence on dementia? For example, having social isolation early in life increases risk for dementia, even if it improves as you age.
So you're asking about whether social interactions early in life, even if you then have more interactions later on, have already caused some sort of damage. Now that's a question that we don't know and we hope to answer, but the studies that have been done so far found that social interaction in late life impact on your cognitive health. So that was just one study. We have data from 12 countries around the world. I hope that we can look at it a little bit more closely to answer that question about at which point is social isolation having the biggest impact.
Okay, so if having someone to confine in reduces cognitive decline, could this role be taken on by a psychologist, and could this be an effective treatment in managing life with dementia?
Yes, I would say so. I mean, I'm slightly biased as someone who used to work as a clinical psychologist, but I would say that relationship with the psychologist is definitely something where you do a lot of confiding. So probably even more than you would confide to other people who are close to you. So definitely having someone regular that you confide in can be helpful. And another risk factor for dementia is depression as well. So looking after your mental health can be very good for you in maintaining your brain function.
Nikki would like to know, what kind of scales are you guys using to measure social interactions?
So the types of scales that are available right now are the UCLA loneliness scales or social network indices, which ask people how many friends, how many family members or they have in their network, how often they interact with them. And then we have some relationship quality scales, but they're different across different studies and in different languages as well.
So there's another question from Yamima. So like, why did you guys decide to focus on brain reserve specifically over the other factors that you listed contributing to dementia development?
So because we are in the position where we have this new imaging data, I think it makes it very exciting for us to look at brain research, but that also means that we're not just looking at brain research, we're going to look in our final study at the mediation of multiple factors. That's your physical health like diabetes, cardiovascular health, your social health, which is your interactions, your environment, and how all of these interplay with each other to impact brain health and vice versa.
I feel like you already just answered the next question, but Linda would like to know, do you consider factors such as diet and exercise as to how it contributes to dementia?
So yes, we are looking at all the factors that you saw there that I mentioned that contribute to dementia. The only ones out of all of those factors that we don't have access to right now are brain injury, because only 2 or 3 studies have given us that data. And the other one we don't have access to is air pollution levels, because people have obviously moved around quite a lot in their life in the last few years. So it's hard to get good air pollution data for those people. But we have access to all the other variables that we know link to dementia, and we're controlling for all of them.
Okay, so there's a question from Nikki. So we'll be looking at how other variables, for example, like having pets, might buffer risk from social isolation.
Someone suggested that actually before, and I think it's a great question. I think someone should look at that. Unfortunately, we don't have that data with us, but it would be a really cool PhD project for someone to look at life with pets and whether that's helpful.
Next question. I'm not sure if Kim would like to be unmuted to ask a question, but we'll go to Yamama first. So she asks, what are some ethical issues that you've encountered with brain research, and how have you overcome that?
So I guess one of the ethical issues that we have is how we store that data. So we have to make sure that we store that data in a very secure format. So using platforms that have been approved by UNSW that are secure, going through a VPN, going through passwords, multiple factor authentication. All of these things are quite important, and only using data from participants who have agreed to pass on their data from these studies. So for example, people who signed up to the UK Biobank said, I actually give you my brain data so that researchers around the world can use this. So we can only use data from people who've given their consent. But these data are de-identified, so we just don't know the identity of these people.
Okay, so there's a question from Rohit. So why did you decide to focus more on research rather than clinical practice?
For me, it was a bit more personal, because when I was a child, my great-grandfather was 90 at the time and was quite active and quite healthy. Used to walk about 2 hours a day. And when we moved house, he couldn't do his volunteer work anymore. And we noticed that his health went downhill very quickly. He developed dementia, and within 6 months, he passed away. So when I was doing clinical practice, I saw a lot of people with dementia. I did a lot of neuropsych assessments and treatments. But when I got this opportunity to work at the Center for Healthy Brain Aging, I just jumped at it because I wanted to have a chance to find out what causes it and try to prevent it at a population level.
I have a personal question I want to ask. So we know that having more social connections helps buffer against dementia. So with the advent of online communication nowadays, do we find that online communication also has this buffer effect?
That's a question that we don't know the answer to yet. And that's because a lot of the studies that are longitudinal started about 30 years ago and didn't have online communication. So that's a question that needs to be answered when we have enough data on that. But I suspect that any kind of interaction might be helpful and better than no interaction, especially if you consider it to be normal interaction. But that's a question that remains open.
So in your research, have you noticed cultural variations in social isolation and about like dementia risk? For example, like in some cultures, multi-generational families, and also like more likely to live together, maybe?
Yes, so we do get a significant effect of the study. So what we're seeing is that different studies have different levels of effect in our models. So some countries show higher effect of social interactions just at baseline compared to other countries. So it's very interesting. What we're going to do next is try to group these countries in some way and see if certain groups of countries show you a different result compared to others.
Later asks, can loneliness become confounded with anxiety and or depression, seeing that those are typically quite comorbid with dementia? And if so, do you separate or control for these variables?
So we do control for depression when we look at loneliness, because we are quite aware that this can be a confounding variable. In fact, social withdrawal is a diagnostics or diagnostic criteria for depression. So we made sure to control for that.
Okay, so I think so unfortunately this is all the time that we have for tonight. So I think like I would say like this is like a really great educational experience, and we're absolutely honored to have you here with us, Dr. Samtani. Sorry. So like thank you for giving up your time to deliver your knowledge in these topics.
Thank you.
And we'd also like to thank everyone who attended tonight's talk, and we hope you found it really inspiring and educational. So please feel free to follow the YNA page and the CHeBA page for updates on future events, as well as to keep up to date with the great work that CHeBA does. So yeah, have a good night, everyone.
Thank you.